So welcome to week 9 of the next generation sequencing analysis course. Um, we've been through a number of applications now in whole genome resequencing. We spent quite a bit of time with the process of alignment and alignment processing, SNP calling, structural variant calling, as well as learning some basics in R and Unix. And we're about to transition now into the world of RNA-seq and studies of gene expression in next-generation sequencing. But before we jump into that next week, I have sort of a grab bag of thoughts and that I thought I would share with you. This week's homework, we are working with genome intervals using and introducing you to the BED tools suite of methods. And so I'll begin the lecture talking about the genomic intervals and some ways in which we go about handling genomic interval data. I'll tell you a little bit of the challenges of dealing with genomic interval data from from a web server and visualization perspective, and then I'll just have a kind of a grab bag of thoughts about data management, the problems with next generation sequencing, with the big data problem, and then just some additional thoughts about programming languages and how they're of use or might be of best use to you as you go throughout your careers. So let's just begin. And I wanted to introduce the browser extensible data format. This is the BED format because this is maybe one of the simpler ways in which we store interval data. And there's one twist with the browser extensible data format that we haven't yet encountered in this course, and that is the subject of zero-based formatting. So this is just an example I pulled here from human chromosome 7, a set of intervals here. This is what's called BED6 format. There's 6 columns. The thing to note here is that the position in column 2 here, this coordinate, it's a 5' coordinate, this is a 3' coordinate, but this is what's called a zero-based coordinate. What that means is that it's zero-based, which means in order to actually this coordinate here actually refers to one base pair downstream of the position that's recorded. That's because it's a zero-based coordinate, so we have to add 1 to the position in this column to refer to position 1 in the genome, so or to the next position in the genome. So in other words, if we had a position 0 in the genome in a BED file, that would be referring to position 1 of that chromosome. Okay, so this is a zero-based coordinate system. Here's an example here, another example maybe where we can see the numbers a little bit easier. This is this pair of coordinates here referring to positions 101 to position 200 on chromosome 1. And in GFF, which is a one-based format, that same interval would be referred to as 101 to 200. So notice that the 3' coordinate here is not zero-based. Okay, so this is notice that in both GFF and BED, the coordinate on the right, or the 3' coordinate of the interval, is a one-based coordinate. So this has some, I just wanted to make sure I knew what I wanted to say here. Why use zero-based coordinates? Well, essentially the reason has to do with the number of mathematical calculations it requires to get the length of an interval. So this is just a little bit of history here of why this BED format was developed. And notice here that SAM, BAM, VCF, GFF, as I showed you, all used the one-based coordinate system for both the 5' and 3' intervals. And if you want to get the length of that interval, you have to subtract the beginning from the end, but then you have to add 1. And ultimately, that addition here, that additional mathematical operation that we have to do in order to get the length, costs computer time in rapid access, you know, applications such as on these large web servers, including gbrowse, where there's a lot of intervals that are being processed very rapidly for visualization and other purposes. So just some notation that you'll commonly see in the literature or in tutorials and things. This is an open bed is an open exclusive or format on the left-hand side, and a closed or inclusive format on the right-hand side. So this is another way of representing that, where the open circle here means that we're excluding the first position in the interval. So the 0, remember, is not referring to the actual position that this interval is representing. Okay, which is why we call it open or exclusive. Okay, so don't let the notation sort of trip you up here. The main point here is just, once again, that you have to add 1 to a zero-based coordinate in the left-hand 5' coordinate of bed intervals. So the bed format sort of has many permutations. Bed 3 is the simplest, so all bed files always have the first 3 columns with the chromosome, contig, or scaffold identifier, followed by the start and end. Then there's additional sort of add-on columns that are possible. The name of the feature or interval, frequently column 5 has some numeric value that, well, it's not restricted to numeric value. It could be a p-value, an enrichment score, but it also could just be raw text. There's no constraint here, but we call this the score column. The strand, so keeping track of strand information, such as which position a read mapped on, or you map to reference genome. Sorry, which strand a read mapped to, or which strand the sense strand of a gene is on, could also be recorded here. So there's lots of good information at the UCSC site at genome.ucsc.edu FAQ page. Has a lot of information on various types of formats, and one that I frequently visit when I have a question about formatting issues, although it's always a good idea to visit the actual raw specification for the format when you are programming and need the absolute details to get right. So just there are additional columns for versatility. Up to typically 12 columns is the most I've ever seen, what's called bed 12 format. So what do we mean by intervals? Well, I think that's obvious, but I think the thing that I wanted to convince you of here is that we can think of essentially any feature in the genome, or even our short reads after they've been mapped, as being intervals. Okay, so really a lot of the processing we need to do can be sort of reframed in terms of manipulating genomic intervals, or processing the overlaps between intervals, or the non-overlaps between intervals. So some common questions here. So here is which features overlap. So if we have 2 sets of intervals, so imagine perhaps 2 separate files, one in blue, one in sort of orange here. Imagine that this is a set of genes and this is a set of repetitive elements here, and we want to know, maybe our question is how do repetitive elements overlap genes, for example. So while that seems like a fairly straightforward question, when you're dealing with millions, or a large number anyway of these intervals, the computation becomes slightly challenging, and you wouldn't want to have to write your own sort of software to deal with that problem. So we'll get to the software in a second, but so how many features overlap might be another question that you might ask. So which features, how many features, by how many base pairs do the features overlap? What is the DNA sequence in the overlapping intervals, and which features don't overlap among many other questions that we can pose about genomic intervals. So this just brings me to the exercise for today and for this week, where I've got you going in and using one of my favorite tools, which is bed tools. It's again a set of command line methods that allow you to do some very sophisticated processing of the raw interval data. So I've mentioned some of these things already. There's a simple command line interface. One of the nice things about bed tools is it reads from standard in if you so desire, and it outputs to standard out. So in other words, we can form long pipes. Now that you guys are getting familiar with that, you can use your familiar commands that you're used to at working at the Unix command line, but you can also now string them together with the bed tool set of methods. So ultimately what that allows you to do is, in very simple command lines, answer some fairly complicated, or what would otherwise be significantly daunting programmatic challenges. So here's one method. This is the intersect bed, and here the Aaron Quinlan here, who's the developer of bed tools at the University of Virginia, has some really nice tutorials, and this is coming from his website on bed tools. Again, you go over this in some detail in the homework, but I guess we can go through a couple of examples. Report the base pair overlap between sequence alignments and genes. So in one file, we specify, for the A file, that's the first file, we specify reads, and for B, we specify genes. Okay, and the output for that, okay, by default, is a set of intervals that represent the overlaps. So one thing that I'll just point out here quickly to orient you, or something to think about, is that bed tools gives you a lot of flexibility in the outputs of intervals. So by default, when you just pass in with intersect bed, here is the program, and there's many programs in bed tools, this is just one. You by default get the intervals that overlap between the two sets of features. Another option would be to say only output the reads that overlap the genes, or vice versa, or maybe you'd like to report all genes by all read combinations. All these are possible. It's just that I think the most important thing when you're working with bed tools and these intervals, which I think for people that aren't used to working with them can be a bit confusing, is to keep in mind exactly what type of output you are interested in to answer the question that you have. So thinking clearly about that is important. Okay, I'll let you go through this. If you want to hit pause and look at some of the functionality here, that's fine. I did want to quickly mention that there's been a nomenclature shift, and so in the most recent release of bed tools, you'll notice that intersect bed, as it's depicted here, is no longer a binary that you download and install. Rather, bed tools now operates with its command called bed tools space intersect, and that's the sort of the new nomenclature that they've adopted for the entire package. So this is a bit old, so don't let that trip you up. You'll be able to see that more clearly as you read some of the documentation and proceed with the homework. Just pulled a couple of other examples here. BAM to bed, while that might seem like a strange conversion, there's a number of conversion functionalities here. So you can convert an entire set of read alignments to bed intervals. So that would be a very large file. Obviously, if you're dealing with a large BAM like most BAMs are, to convert that to bed would be a lot of text that might be undesirable, but people have their reasons, simply because sometimes softwares require various inputs, and if a software requires a set of intervals and you'd like to and won't accept BAM, it's a good chance it might accept bed. So I'll just leave these. These are for you to consider. These are additional command line programs. I just wanted to give you a sense of just how flexible this is. Closest bed is an interesting one. So for example, someone asks you to identify the set of SNPs that are closest to a particular feature, say the 5' position of a gene, or maybe are closest to a transcription factor binding site or enhancer element in intergenic DNA. That's a common type of question, and so you could use closest bed to sort of quickly pull those sets of features out. So this is coming directly from the Quinlan and Hall paper. I put this on the course site for your for you to take a peek at, and this just gives some of the functionality that was present a couple of years ago when the paper came out, and they've added additional functionality since then. So oftentimes, if you have a simple question about overlaps, intervals, counting them, identifying sequences of intervals, so like FASTA from bed, so you can extract a DNA sequence out of a FASTA file quickly for a set of intervals. Coverage you can extract quickly. Many things you can do here. Sometimes I think it's a good idea to just be aware of the flexibility of a set of tools like GATK or bed tools or VCF tools, because this is how you expand sort of your bag of tricks that allow you to work efficiently and comfortably with these large data sets. So another place where intervals come in and are particularly important and how they're handled and is in the visualization and maintenance of large data sets on web-based servers. So here the premium is placed on access time and the ability to display a lot of features rapidly, and to adjust with the display with the click of a button, etc., etc. So I just wanted to show you an example of that. This is some expression data. These are tiling arrays. So tiling array data is coming from just like a microarray, except that tiling arrays have probes that are sort of evenly spaced across the entire genome, not just in known transcription areas of the genome that are transcribed, such as exons, but tiling across the entire genome to try to discover new transcripts that may have not been already annotated in a genome. And so this is really dense data, and so this is a case where people developed a special format type, such as the Wiggle format or WIG format, which is good for tens of millions of points with regular intervals, and other sort of like bed graph, which uses some of the features of the raw bed format to display less dense data. So once again, this is bed graph here. This is just not, gives you an example of what bed graph format looks like here. Notice again the zero-based bed intervals here with values in this case in the fourth column. So this is a slight deviation from the format that I gave you before, which had a name here in the fourth column, I believe, not a score. But this is okay. I mean, bed is flexible in that way. So I just thought I'd switch over now, switch gears, and talk to you about some of the issues in big data, data management, and ultimately the programming interfaces or programming languages that we use to handle these big data, among other issues. So this is kind of a grab bag here, a few thoughts, things that I felt like we've been in the course for almost 10 weeks now, and there's some issues that we just haven't mentioned. And while the focus of the course is in expanding your toolkit, giving you the skills that you need to begin to work with these large data sets, we should at least talk about some of the issues that are involved that you encounter as you go along. So this is I thought I'd start here with a map of these sequencers, the next generation sequencer distribution across the planet. So here you can see 306 sequencers in the northeast of the United States. That's heavily skewed by the Broad Institute at MIT. We have I think we're almost to 100 here in New York City, but I don't think we're there yet. but the New York Genome Center is starting to, or will bulk up the number right here in New York City. You'll see 7 over here. This is where I have a close affiliation with my colleagues at NYU Abu Dhabi, and so I actually added our facility there to the omics map over here, representing our HiSeq and our PacBio and other machines that we have there. So check it out. This is a typo. Sorry. It should be omicsmaps.com. So there should be an S after omics. If you want to check it out and see where the sequencers are nearest to you, you can zoom in, zoom out, and get a sense of the density of sequencers in the area. So really, there's a whole new field. Many of you are obviously aware of the field of big data science. I've been part of a panel at NYU where we have been seeking support for our big data science program. And there's a lot of issues that we talk about in big data science. Some of them I'm mentioning here. So in the context of next generation sequencing data, the data production rates are kind of the astonishing thing. Roughly about 2007 or 2008, things really shifted. So people use sort of Moore's law as a comparison. This is the rate at which computer processing times increase here, and so we're actually showing it sort of the inverse of that here to fit with the cost on the y-axis here of generating a certain number of base pairs of sequence. I don't know what the axis here actually is, but the point here is to just show that the sequencing acquisition increasing, or the decrease in cost of sequence acquisition was roughly related to Moore's law, but then in 2007 there was a jump in technology, obviously the next generation sequencing technology that led to this massive departure now. We're collecting far more data, and there's this rate of increase here as the technology took off. And just to throw a number out there, we're producing, this is as of 2011, was the most recent number I could find, was 13 quadrillion DNA bases a year. That's a quadrillion. I had to look what that is. It's 1 million gigabases. Okay, so 10 to the 15 base pairs per year. So this creates a massive bottlenecks in storage, processing, software engineering, or creates a need for the software engineering and a need for data provenance and provenance management. So I thought I'd give you some numbers here for things like you can expect, like when you're working with this type of data. A typical lane on our HiSeq, we're getting about somewhere between around 20 to 30 gigabases of sequence per lane now, and in the end, what I just did a quick sort of back of the envelope calculation, and to store the raw data alone in fast queues, you need roughly 1 byte per base. Okay, so if you have 30 gigabytes of sequence, you need 30 gigabases of sequence from a lane. You would need 30 gigabytes of storage. Okay, so many of your laptop computers are probably somewhere, nowadays between 100 gigs to 500. Maybe if you have a really nice laptop, maybe as much as a terabyte now. But so this is what you can kind of expect now from typical data set. I usually tell my boss that we need a minimum of 100 gigs for the data sets that we collect. But when you're processing the data, you're oftentimes generating a lot of intermediate files, a lot of things that eventually get discarded, but that you need in order to work with the data. And so on our computer clusters, we have access to about 5 terabytes of data, of storage with which we can work with while we're processing the data. So this far exceeds what you generally would have on any kind of personal computer, and you really need sort of high performance disks to work efficiently with this volume of data. That's not to say that personal computers don't can't get the job done for certain scales of project, but for the really big projects and at large funded by large universities or by governments, you generally would need significantly more storage space than you would have access to on a personal computer. The data processing, typically our workflows require clusters, or in some cases even grids of compute nodes. However, I thought I'd throw this out there that for a lot of applications, it doesn't make sense to maintain all of the nodes that you would need in order to work with this type of data. And I posted a paper on the course site about cloud-based computing, simply to introduce you to the concept. I'm sure many of you are intimately familiar with it, but there's lots of sort of innovative ways people are thinking about applying cloud-based computing to next generation sequencing problems, and making available different types of cloud-based resources. So one problem is in the sharing of software, the mounting of software onto these remote clusters. So that people have, for example, come up with ways of developing entire images. These are sort of suites of software packages that can be immediately loaded for a user, so that they can work in their environment that has SAM tools, GATK, all of the tools that we've been working with in the class, and immediately begin to work on them in some remote server somewhere. There's also issues with how to pay for these resources. So there's lots of clever ideas out there. So one interesting one mentioned by the paper that I'm familiar with the Amazon EC2, is the idea that which is one of the first cloud-based, commercial cloud-based compute resources. So the Amazon EC2, you can go in and bid for compute time. So you can, they might have an extra set of nodes. They want to make money off those nodes rather than just have them sit idly. And so you can go in and bid and say, well, I'm willing to pay, $1 per hour for a processor or what have you. And, then if they are agreeable, then you can go in and have your node for $1 per hour. So there's lots of need for software engineering and essentially taking processes that formerly we just were developed for serial processing. There's a need now to parallelize jobs to make them tractable for or to make it tractable to complete the job in a reasonable amount of time. So there's a huge move now to make software multi-threaded for that purpose. Finally, I just thought I'd mention this is an issue that I feel is important in big data sort of management, is provenance. So for imagine librarians, or excuse me, let's take for example, not a librarian. Let's take for example a museum where there is art. And you go to the museum, and people want to know who owned that piece of art, where did it come from, when did it change hands. When you buy a piece of art, that's oftentimes the first thing you want to know, is who's had that piece of art and where did it come from, and ultimately who painted it, and anything that's been done to it in the process of acquiring it. So this is something that's important in data management. Like, we'd like to know, when we have a set of SNPs, we'd like to know, where originally did the reads come from, what are all of the different tools that have been used to process those reads, and eventually, what SNP caller was done, what genotype likelihood model was used, And so all of this becomes important because in the end, when we're submitting all of these data to clouds and we don't know what's been done and there's multiple people working on the data set, so it's not clear who did what or when was what was done to the data, what I can tell you from working with this type of problem, that it can become confusing and can lead to sort of decreased sort of confidence in the quality of the data and the data output. And so one of the things that people are working on is ways in which we can track the provenance of data. So I meant to show you this a couple of slides back when I was talking about the compute nodes. So this is just an example of our cluster, a visual display of our cluster as it was about 20 minutes ago at NYU. This just is a display that shows you our capacity at NYU, which we really need in order to work with this type of data. So we have at NYU about 230 nodes. So I'll just give you an illustration here. This block of dots here, if you've never worked on a cluster before, is a node. Each dot is a core or a processor. And so we have about 230 nodes displayed here and about 2,500 cores. Okay, so in colors here are different users and their jobs are running. This user here has 8 cores, then there's 4 additional cores not being used on that particular node. You can see that many nodes are shared among users, which also creates its own issues because a different user can crash out your node because they requested too much memory or something. And so sometimes this introduces its own set of problems in working on shared resources compared to when you're working at home on your own computer. And if you crash your own job, then you only have one person to blame for it. Anyway, so let's go on. Quick note on databases. I didn't feel like I could go through this class without mentioning it. This is something that I'm hoping you're getting in your other bioinformatics classes, because we won't have an opportunity to really talk about it here. It's really something that only we use intensively sort of in the latter stages of data processing. In fact, storing short read data in database simply wouldn't work. It's too much data to store in a database. So after you've condensed all of the data down, you've called your SNPs, you've identified your interesting mutations, you want to relate them back to the genes that those mutations are in and what the functions of those genes are, and what the, for example, PFAM domains, what the interpro predictions are for the function of a particular gene, et cetera, et cetera. We want to relate all of this, cross-reference this different types of information. And so this is where databases start to become really handy. So the simplest form is flat file or a tabular file. We've been working with those in our working with R. XML is important, particularly when you're working with databases that are online, which oftentimes dump their contents in XML. And of course, relational databases like MySQL are of paramount importance, and if you get an opportunity to learn MySQL, that's a marketable skill, one that's important not only in bioinformatics but is important in many fields. So let's move on to a set of projects that I think are important in hosting a lot of the data once they've been processed to some degree, like the genome data, the genome browsers, now the comparative genomics browsers. Soon I expect there's going to be many resequencing based browsers that host all of the polymorphism or SNP data that is being collected. And one of the probably most developed set of tools for handling all of this data is the GMOD database project. So the general, the generic model organism database project. It's a collaboration initially between Flybase, Yeastbase, the Mouse Genome Database, and Wormbase, where they got together and said, hey, we're not going to develop all these tools independently of each other because that's a lot of redundancy. So let's get together and make a cool set of tools that we can all use to host our genome data. So there are now this whole Gmod project. You should go to gmod.org and check it out just to familiarize yourself with the flexibility of it. But its modular design really allows you to customize the database for how what the contents the database holds, how it's displayed, etc. And so here's some of the components. Maybe you've heard of some of them, like gbrowse. Virtually all of the major database organism databases have a version of gbrowse, so that is a component of the Gmod project. So you could go to, say, TAIR, T-A-I-R, and that's where the Arabidopsis information resource is, and they have gbrowse there. You can go to ones that I work with every day, like Michigan State University's Rice lab at Robin Buell's lab at Michigan State. They have gbrowse there on their server. You can go to Phytosome. I believe it's phytosome.org. That's where all of the plant comparative genomic browsers are. That there's probably 30 different species of plant genomes data that are hosted there. Anyway, many mouse genome database, mammals, Encode, Gramine, many other databases out there that you will find gbrowse. Pathway tools, maybe you've heard of BioPsych, PlantPsych, EcoPsych. These are databases that whose focus is on network data, such as metabolic networks, regulatory networks, etc. And then one thing that I wanted you to, we just have to mention it, is the galaxyproject.org. So while in this class our focus is on developing pipelines, learning methodologies for analyzing data mostly from the command prompt and writing shell scripts, and if you have the skills for writing computer code, the Galaxy project provides an interface to a lot of these tools that's a graphical one. Okay, so this is a viable model. There's a lot of people out there that will never learn the bioinformatics skills that you guys are developing, and they want to work with this type of data, and you can. I mean, there are ways to go about it. It's not my personal preference because I feel like when I'm working at a terminal with and just working with a graphical interface, that I'm sufficiently removed from the actual things that are happening behind this under the hood there, that I feel a bit uncomfortable and I can't track what I've done. This is part of the provenance issue that I mentioned before. It's important to a lot of bioinformaticians to be able to track what we've done. So to provide a shell script, for example, to a colleague to show them that this is exactly what was done to the data, etc. But this is a viable model, and it's probably the most advanced and most sophisticated of any of the graphical-based analysis suites that are out there. So this is my attempt at using Gmod here. I'm currently working on the 100 Dates project, as I think I've mentioned before. We're sequencing 100 different date palm varieties, and me and my colleagues at New York University, Abu Dhabi. Here's this, image of gbrowse that I, took, just a screenshot from my computer, where in a matter of about 2 days, probably would have taken me less than that if I'd really worked hard at it. You can go from basically nothing to at least having some ability to display your data. So there's a lot of, I'll try to wrap up here. There's a lot of bio-community based projects. This is sort of something that you can even get involved in without knowing a lot about Perl or Python, Ruby, Java, etc. So many of these projects are object-oriented projects, so you can go in and do an enormous amount with knowing very little, and that's because there are snippets of code that you can rip from these sites that will perform the tasks that you would like them to perform, as long as you are convinced that it's doing what you want it to do. The code is frequently available for it to be shared. That being said, ultimately you guys will want to pick up one of the scripting languages, either Perl or Python. And in the future, maybe we can chat about that. Where some advice. I have some advice that I could probably share with you. That might help you in making a decision if you haven't already made a decision about where to invest your, important time in, either Perl, Python, or possibly Java even. Though that's more for software engineers and developers. So Perl and BioPerl is really important in CGI-based web interfaces, and much of the Gmod project. In fact, virtually all of Gbrowse, anyways, is Perl-based, and I believe much of Gmod project is Perl-based, and a lot of BioPerl went into the development of Gmod and other biological databases. Of course, BioPerl has a lot of overlap in these other tools with BioPython in particular for doing a lot of the common day-to-day activities, things that we need to do sort of in processing large data sets in bioinformatics. Some of these things will look familiar to you. It's very modular, obviously, so that's one of the beauties of being able to write your own code and to be able to use shared resources like BioPerl. You don't have to reinvent the wheel for doing single tasks, but then you can string together many simple single tasks into making quite complicated pipelines to analyze data. So BioPython is also fairly mature. BioProject, BioRuby, and BioJava are far less mature in their development. Bioconductor, we talked a little bit about that. I believe they're up to over 700 packages now at Bioconductor. So oftentimes if you have a need, say I need to analyze differential expression and RNA-seq data, Bioconductor is a way to go. One of the drawbacks of Bioconductor is that each of the packages sort of has its own set of functions, things that have to be learned individually. So one of the reasons why I've sort of hesitated to go in and do a lot of individual Bioconductor packages is because you end up learning things that are very specific and not necessarily tools that are generalizable. So I don't want to have you guys spending a lot of your time learning things that you may never use again, whereas I'm quite confident that learning just the R statistical language by itself is probably worth your time because you will use it again. But anyway, I think we'll go in and take a look at DESeq, which is differential expression package in Bioconductor. We'll take a peek at it in the next couple of weeks when we look at RNA-seq data. Biomart, just be aware of it. This is a portal for accessing many databases that are it's a unified portal, so it's a way of integrating sort of many different independent databases and accessing them all through a common interface. Very important when you're wanting to analyze a diversity of data types from different organisms that all may come from different databases. This is one way to sort of unify that analysis. And then finally, I just thought I'd throw out there the BioCamel project. So this is something that is actually NYU has had a lot to do with. Ashish Agrawal, who's actually just now left NYU Center for Genomics and Systems Biology, is actually the lead on this bio project. It's a functional-based programming, which if you're not familiar with, OCaml is the language. This is it's a language or a programming paradigm, really, that's based in set theory. It's relatively new, although it's very popular now amongst computer scientists, simply because of the way it allows you to generate abstract code that for end users like you and me, they promise will revolutionize how we handle our data, simply because what they argue is that they can make program sufficiently abstract that all you would need to do is say, this is my input data, here's what I want to do with it in a simple way, and then they can provide outputs without having you have to worry about all of the management of the data, like data format conversions, for example. So this is something that there's a future in functional-based programming, in next-generation sequence analysis. I think it'll take a long time for it to sort of take over the way in which we currently process our data, but it's something to take away from here, just as something to keep in the back of your mind that there's something else going on out there. And if you're curious, go ahead and take a look at the BioCamel page or OCaml. All right, and with that, I think I've talked quite enough for today. So I guess I'll see you guys next time.